in the brain cells in your brain. The nerve cells in your brain don't divide, right? They're amitotic. They don't have the cellular mechanics to divide. But these neuroglial cells do divide. They divide pretty fast. And when they do divide, and if they are mutated or obviously cellular machinery goes wrong, they can cause major tumors. So ionizing radiation and certain other diseases can cause the onset of these gliomas. So, oops, there we go. Astrocytoma, obviously cancer of the astrocytes. So this is a much more aggressive form of a glioma. So usually these tumors are treated pretty aggressively with surgery and radiation to help remediate. They can get very dangerous because what they're doing is they can compress the areas of the brain that are normal functioning, right? So just because the normal tissue isn't affected doesn't mean it's not going to be squished or like frozen out, by these huge tumors. And everything's encased in your cranium, so it can be very debilitating. Okay, so now we're going to get into the electrophysiology stuff, right? This is how they conduct. We understand the organization, structure of a neuron, how it all is put together, all of the key players. This is going to sound very similar to muscle physiology, because in muscle physiology, we did some neurophysiology. We've already laid the groundwork for some of this stuff. That sodium, potassium pump, the leak channels, all those things. It's coming back to revisit us. So if you didn't get it right the first time, here's your second chance, all right? So all neurons are excitable, right? They have this ability to become excited and do something, just like our muscle cells. Skin cells, not so much. Muscle cells, yeah. Nerve cells, yeah, right? So they can respond to several stimuli, just like our muscle cells, right? Respond to the stimuli, electrical signals, chemical signals, mechanical signals, right? Remember those different gated channels? There they are, electrical gated, chemically gated, mechanically gated, right? So they're meant to conduct these impulses rapidly, right? Nothing in our central nervous system is about slow and steady. Everything in our nervous system is about get it done, right? Communicate this quickly. So, two ways these electrical currents form. One's called a local potential, and one's called an action potential. A local potential would be one that travels a short distance. So, a local potential might travel, let's say that there's another neuron here, right? And it's communicating with this cell. It would cause a local potential right there. A local potential. It's just a small area that says, oh, hey, someone just tapped me on the shoulder, right? The local potential is just a small change. An action potential is where this change is going to go down the entire length. So guess what? If there's enough of these local potentials, our axon says, hey, enough tapping me on the shoulder. What the heck do you want, right? It's going to basically sum all of those signals and use it to transmit its own signal. So that's the action potential. So this has to do with this unbalance of positive and negative charges across the membrane. Same general setup as what we had before, right? We have an overall negative potential inside the cell, an overall resting negative charge. Outside the cell, we have a positive charge. We have the same distribution of ions. We have more sodium outside the cell, more potassium inside the cell. And so this creates that negative resting membrane potential. So voltage, let's just get some general terms out of the way. Voltage is an electrical gradient. Voltage is the difference across the membrane. So when you have a difference, you have a voltage, right? That idea of a potential, that charge, is a voltage. There's a voltage on the membrane. The membrane potential is that electrical potential across the membrane. It is what's the difference between the inside and the outside. Typically, in our neurons, we're looking at a resting membrane potential of -70. Right, -70. So our cell sits at -70 under normal resting conditions. Let's review all of our mechanics just in case. I'm just going to draw a regular cell. Our cell likes to have lots of potassium on the inside. A little bit of sodium. Lots of sodium on the outside. All of these are ions. I'm lazy and not labeling them as positive ions, but they are all positive ions. Our cells have two types of channels on them, gated and leaky. Right? The leaky channels, I'll make them blue. They can leak out potassium. This is going to be a potassium leak channel. A sodium leak channel is going to leak sodium in. This is all the same exact stuff, right? And then we have gated channels. Gated channels are specific. They're only going to open and close under specific conditions. Gated potassium, gated sodium. There's also our pump, right? Sodium potassium pump. Our sodium potassium pump is doing what? What's it pumping where and how many?
3 down, 2 in.
3, so what's being pumped in?
So it is,
What's being